Hello and welcome to this live webinar hosted by Lentivirus. I would like to welcome you all to this meeting today. So this series of webinars we host specifically for CMC developers on analytical challenges for biologics development, and of special focus is HCP and analytics for HCP analysis. And so welcome to this. I have also today's speaker. Welcome, Victor.
Thank you.
So before Victor gives his presentation, I'll just talk a little bit about practical things for the webinar. So the talk will take about 30 minutes. I will give a short introduction to the topic and HCP analysis in general and what is driving the industry right now. And then Victor will dig into the science, which today is about bioinformatics analysis of HCPs using mass-spec data and what we can learn from this. If you have a question during the webinar, then please write them in the chat. And then there will be 10-15 minutes for live Q&A after the presentations. So yeah. With that, I think we'll just go live with the slides. So let me just start the slide presentation. So I hope you can see the slides now. So yeah. So what is driving the industry? Right now, there are some major events and major improvements in technology. That is the cause of why we are talking about this topic at all. So let me just see here the first slide. So the first and most important event is a new USP general chapter named 1132.1 on measurement of residual host cell proteins in biopharmaceuticals using mass spectrometry. This chapter is in addition to 1132, which describes the use of ELISA and ELISA reagents for whole cell protein analysis. So this chapter was the first draft released in May last year, and USP has now received a lot of comments for this. So the final version of this chapter will become public 1st of December this year, according to the US Pharmacopoeia. And the chapter will become effective from May 1st next year. So in addition to this important publication, there are a number of key publications for HCP analytics that you should be aware of and that are driving these changes. One important paper is this paper by Rosalind Moulton and colleagues from Regeneron, where they characterized the host cell proteins in commercial therapeutic mAbs. So they characterize all the impurities in mAbs on the market. The second publication was from Martin van der Laan and Kevin van Cott and colleagues. It's a review paper on HCPs observed in clinical trials that stopped the clinical trials or delayed them significantly. So they collected all this combined experience also from non-published work. And so this is a really great paper on what can go wrong in different types of biologics due to specific host cell proteins. The third important paper is a collaborative paper by the Bioforum group and companies collaborating in the Bioforum. So where they collected a lot of HCPs of potential concern and published this both in this paper from 2021 and also in a searchable database that you can find at the Bioforum database. So these publications and technology development, the industry is really driving changes in how we look at the HCPs in biologics. So just to read some things from the USP chapter, why these changes are needed. So the first page in the new chapter describes the limitation of HCP ELISAs. So I'll just read up here. First, polyclonal antibodies used in immunoassays often have no or limited coverage to particular HCPs that are non-immunogenic or only weakly immunogenic. Secondly, the immunoassays may underestimate or overestimate levels of HCPs if HCPs are non-immunogenic or they are so-called high responder HCPs with antibodies dominating the ELISA. Thirdly, the ELISA will only generate a single value for total HCP amount, and it does not distinguish between the contribution of the ELISA signal from any individual HCPs. And lastly, the chapter describes that the lack of specific identification of individual HCPs prevents a knowledge-based risk assessment in the clinical trials and for product stability on particular HCPs. So that is the reason why the USP expert panel wrote this new chapter. If we look more into details, what the chapter contains, it has a description on best practice and analytical methods for the use of mass spectrometry. So it describes the sample preparation, how you do protein digestion of the HCPs, how you do chromatographic separation of the peptides before the mass spectrometry analysis. It describes different mass spec setups, and then it describes 3 different principles of how you can quantify or relatively quantify the HCPs. There are 3 different methods you can apply for quantification or relative quantification. The first one is you quantify the HCP peptides against drug substance peptides present in the same sample. The second approach is to use isotopic labeled peptides for the specific HCPs and spike these into the sample after the protein digest. And the third approach is to spike in intact protein standards and then quantify against the peptides from these standards. So all the 3 methods are described in this chapter, and we will show you data from one of these approaches. So with this new technology and the new chapter, it opens for changes in the analysis strategies you have for HCPs in your clinical development. So the old way of doing this was to use ELISA only. First, you would typically use a commercial ELISA, and then you would shift to a process-specific ELISA once you locked your process. But with the advent of mass spectrometry methods, most companies today are using mass spectrometry as an orthogonal method to guide process development and to look into specific HCPs that may be problematic for your process or your product. The third approach is actually using mass spectrometry to characterize your ELISA reagent also, and thereby you can show that a commercial ELISA kit and reagents might be completely fit for your process and product. And many companies are now using this approach for also marketed products. And the last possibilities that opens up with the new chapter is to use mass spec only for HCP analysis. So right now we see this for products where it's not possible to develop an ELISA or where you have fast track status on the product and you don't have time to develop an ELISA. So yeah, we can talk more about these in another webinar. So just before I hand you over to Victor, I'll talk a little bit about the principles of the methods that we use here at AlphaLISE and an example of a data set for one type of sample. So the analysis here, it contains 3 different steps. First, a sample preparation, where we spike in these intact proteins, and we use these as standards for QC purposes and internal calibration standards. Then we have an LC-MS analysis that's optimized for identifying and quantify the low level of HCPs, even though we have high amounts of the drug substance. And the third step is a data analysis, where we do a database search to identify the proteins in the sample. So the data analysis, we set up a sequence database that contains the drug substance amino acid sequence and then all the theoretical proteins that could originate from the expression system. So we have used this generic method the past 5 years and provide this as a service. So we have run lots and lots of projects and lots of different types of samples. So we have listed some of them here. So you can see a variety of different expression systems, a variety of different drug types, from most simple recombinant proteins to very complex cell and gene therapies, and also bacteriophage products. So all this using the same analytical method and the same 7 standards. So just to show you a typical data set, so this is from analysis of samples from mAb purification. So we have 4 different sample types: an early harvest sample, a protein eluate, one of the polishing step, and the purified drug substance. So in the data set, you can see list of host cell proteins, their names and accession numbers, so you can look them up in a database. You can see the number in here represent the amount of HCP calculated as a nanogram HCP per milligram drug substance, or parts per million. So Victor will come back to a little bit how we quantify this. So we get the number of the and the amount of HCP. So you can look at the individual HCP and see how it purifies and clears through the different process steps. And also you can see the sum the HCP amount, so you get a total amount of HCP in that particular sample. So here at AlphaLISE we have lots of these data sets, and so the question is, what happens when we have many of these? And that's what Victor will talk about. So I'll just hand you over to Victor's presentation. So please go ahead and dig into the science, Victor.
Thank you, Ivan. So yeah, my name is Victor. I'm something which is a hybrid between a computer scientist and a protein scientist. I'm a bioinformatician, and my expertise is specifically data. So why do we need a bioinformatician for looking at host cell proteins? Well, if you look back just 10 years, looking across 500 ELISA kits, I definitely wouldn't have had a job in this industry. But due to the growing support and use of mass spectrometry, this need has been created within HCP analysis. And mass spectrometry has made it possible to look at individual host cell proteins in each sample instead of just looking at total protein concentration. So this creates a lot more data, which is why we need bioinformaticians for looking at this. And my aim with this presentation is to give you an insight in how we are moving forward with this and applying learning from all of this data. So where did all this start? So when I started at AlphaLISE about in 2020, and I met Thomas and Ivan for the first time, they told me about this robust and reproducible host cell protein analysis they were doing, and that they'd analyzed a lot of different samples. So that generated a huge amount of data with this method, and all of these samples have been spiked in with the same 7 intact protein standards, according to the method described in 1132.1. So Thomas was quite certain and had a vision about being able to use these protein standards to look across all of these samples somehow. And that's kind of where I came in. However, I was actually quite skeptical, because coming from a university setting where we have been working with LCMS data quite a lot, it was notoriously difficult to actually look across and compare different things within these samples, because it's quite difficult to work with, and there's lots of variables, and it's often difficult to recreate your results. So yeah, how can we use all of this data to learn, and what can we get out of it? So from this, we actually set out to create an initial database just containing the 10 different samples coming from 10 widely different projects and organisms, just as an initial pilot experiment. So here is I'm going to give you one of the first looks at some of the figures I presented to Thomas and Ivan to start with from this initial database. And here we have some box plots where you see the total sum-all intensity for the 7 standards in each project over time. And as you can see, there's quite a bit of variation across these 10 projects. And it's also one of the reasons why it's so difficult to quantify with mass spectrometry. However, once we normalize against these internal protein standards, this is a completely different picture. And it shows that we can actually account for this natural variability we see in mass spectrometry. And in this case, it's the quantified values of the 7 intact protein standards per project. So we actually have ppm on the y-axis here. Or we can also look at the individual protein standards and compare them across the quantifications across these 10 projects. And in this case here, we get a sense of the precision or variability in this data. And if you look above, for example, at the laser pointer here, you can see we have these numbers here. These are the inter-assay or inter-CV for each of these protein standards. And I'm certain many of you know what a CV is, or a coefficient of variation, but anything below 30 is generally very good. And especially when you consider this is measurements between 10 completely different projects. So this was an early indication that we actually had a very, very robust and precise quantitative LC-MS method here. So from this, we actually set out to put on a full-scale database using all of ELISA's data. So what do you get if you combine 2,000 plus samples, the 7 internal protein standards, and a robust and reproducible quantitative LC-MS assay? Then you get AlphaBase. So AlphaBase is a SQL database which consists of all of this HCP data we have collected at AlphaLyse. Not only does it contain the HCPs, but it also contains contaminants, standard proteins, and all of this is on the protein, peptide, all the way back to the ion level, and we actually also all the way back to the raw data. So it's a huge database with a lot of potential, a lot of data, and it's structured in a way where we can ask all of these questions to it. And just above here, we have some overall statistics showing just the sheer size of this database and how much data we actually have in it, covering over 40 million ions, total measurements. So that's quite a significant number across 49 different organisms. So how does this compare to what we actually saw before? So, or to the 10 samples we saw before. So here again, we have a figure showing box plots for each of the 7 standards, whereas the internal sum-all intensity, the sum-all intensity for each of the 7 internal standards presented in box plots for each project over time. And as you can see, there's quite a lot of variation, and this is just the natural dynamics of this instrument, and it shows that's one of the reasons why it is so difficult to actually quantify with one of these methods. So something has to be done before you can actually use this data. So what actually makes LC-MS analysis of HCPs so challenging, and what is one of the sources of variability, as we saw on this previous slide? So first of all, we have sample variability. That's one of the main causes, of course. Is your sample homogeneous? Have you stored it correctly? All these things. There are many sources which can come up there. Sample prep variability. So do you prepare your samples in the lab correctly? Are you introducing any sources of variability? Have you automated your process? Has the digest been done correctly? All these things. So there's so many things which you have to account for here, make sure you have control of. Then there's the LC-MS analysis variability. So is the system you're running this on stable? Are you sure that it's running optimally? Because it's the very, very sensitive instruments, so many things can go wrong, and it's also why it's very important to monitor these things. Also, specifically with HCP analysis, we're working with a very large dynamic range. So some of our samples, they'll be have very high protein concentration due to the drug substance in the sample. But what we're actually interested in is like a needle in a haystack somewhere hidden in the shadows of this drug. So we're working in a very large dynamic range because we can't just overload onto the LCMS instrument. And then there's this manual handling and evaluation element. So as soon as you're handling any sample or anything, then you have the opportunity to introduce errors or all of this stuff. So this is also something you really need to consider and automate as much as possible. And then lastly, there's the data analysis variability. So all these different search engines, if you use one search engine or another or a different version, then there's the chance that you will get completely different results, and you're almost certain you will do that. Also with for example, different databases for protein sequences, depending on which one you use, you will also get different results. So it's very important to be consistent, use version control of these different things, and have a automated and robust pipeline for this analysis. And these are just some of the general problems which are associated with LCMS analysis of proteins. So we have an extra element which is due to the vast or largely different nature of the samples we're analyzing. Some of them are very clean drug substance, or maybe harvest samples, cell and gene therapy, antibody. So they can be very different. So we're working with many different types of samples, and we need a robust method to cover all these things, and also to make sure that we don't introduce any unnecessary variability anywhere in the process. So that's something that, of course, AlphaLyse has worked a lot of time to get control of, and that's also why one of the reasons it has gotten to this point where we can use this method as we are. And as I'll show you on the next slide, this is what I can say one of the results of being able to control of this variability. So here we have the quantified values for each of the 7 internal protein standards across projects over time, and as you can see, they line up very nicely. However, there are some where they actually are, we can say, outliers, where they don't line up just as the other ones. So it actually allows us to actually identify when things have not gone as they should have. So with this, we can account for, using these internal protein standards and normalizing against them, we can account for this natural variability of the instrument, and we can also identify when things are not performing as they should. So we can again look at the individual proteins, individual protein quantifications from across all of these 2,000 projects. And in this case, each of these small dots actually represents a quantitative value from one of those projects. And this gives you an overview of just how little variation there actually is across these 2,000 projects. So within let's take a glass of water. So across the 2,000 quantifications, there's a variation, or a CV, of 30%, which is actually quite insane when you think about it. For this AO2, it's only 20%. And in this case, this is comparative to the CV we saw on one of the first slides I showed you, where it's the inter-CV, which is across different projects. The blue is between the different replicates. So this also shows that even between replicates, it's very reproducible. But when you compare it across different projects, it's also very robust and precise. So one thing here is there is some variation in the accuracy. This is what generally within what we would say a twofold change, and this is something to do with the different physiochemical properties of the different proteins. So some proteins will fly better than others. For example, protein number 5 here it has a different ionization energy compared to AO6, for example. So there will be some variation in the accuracy, but the important thing here is not necessarily the accuracy as long as it's within a certain range, but it's the actual precision of this, because it will perform the same relative to the internal protein standards we're quantifying against every single time, and only vary by, as you can see, 23% across these 2,000 samples, which are vastly different. So it just shows how robust and reproducible this method actually is, and these proteins, they reflect what you would expect in your HCPs. So they show how precise these quantifications will actually be for your host cell proteins as well. And in case some of you are wondering, one of the most the protein standard we use primarily for one of our quantifications is beta-lactoglobulin, just to share that with you in case you were wondering. So what can we actually learn from this host cell protein database? So we have all of this data, which is shown to be very precise and reproducible, and we can compare this across different projects and actually learn on these individual proteins. And I'll show you some different case studies and some different examples that we have used it for. So one thing is problematic host cell proteins, and specifically in mAbs. So we have all these, let's say we have 2,000 samples, and of those, 645 of them are mAb-CHO based, and we have a internal database of 1,600 potential concern HCP accessions. And when we search those against our database, we actually identify in total 340 of them in all samples. But if we just look at DS-CHO based, then we find 118. So it's actually quite a large number. And we can dive into this some more and look at, let's say, the top 3 mAb-CHO based problematic HCPs, which we have observed in our database. So in this case, it's what's it called? A statistical overview of the different projects we have observed. So this gives you an overview of what we actually can extract, and one thing you can kind of look at is you can explore different purification profiles. So you can look at how different methods have been able to clarify these different proteins. You can compare different relative levels. So you can see we've seen this protein here in 30 different samples, and the median ppm was 19. So you can get a kind of comparison of how is the level of protein in my sample compared to this. Is it higher or lower? How does it correspond to it? And we can also do this with just the top 20 problematic HCPs, because we know a lot of you are quite interested in these things. So and here is just across all of the different projects. And yeah, so one general thing we've actually used this for was that we had a client which was what's it called? Which was had found some, what's it called? PLB2 in their DS final sample at a level of 70 ppm, and they approached us and asked, how is this level compared to what you would generally expect, and what else you've seen? And because we had this database, we could give them quite a clear answer and say that, well, based on these numbers and what we've seen in similar samples before, we could say that this is actually quite a high level compared to what we would normally see. And because of this, they could go and change their process and bring this down to a level well underneath this median ppm level here. So they were successful in being able to do this. So another thing is just generally most common host cell proteins in some of the different organisms, because I know a lot of you are using quite different model organisms for this, but these are some of the main ones. So we have HEC239 or 93 and CHO and E. coli, and these are just some overall statistics from all of the samples and but it just shows how much different information we actually have in this database and some of the questions we can actually ask to this. So it's essentially only your imagination which actually sets the limits to this, and we can extract a lot of different things from it. So here we have a specific case study of how we actually use this database initially for selecting some polysorbate degrading post-cell proteins for an assay. So we were approached by a client which was struggling with product stability, and they were suspecting it was polysorbate degradation, but they weren't sure about what polysorbate or lipase this was, and something that typically other mAb developers also experience. So based on their experience and also this recent paper by Daniel Kovner et al., we could create a list of potential polysorbate degrading lipases which we expected could be the problem. And from this, we could apply this to the database and cross-reference and see which ones we actually were detecting to see which host cell proteins, in this case, were actually a problem in real life. Because we had a huge list of these lipases, but we wouldn't be able to make an assay covering all of them, and it wouldn't necessarily make sense either. But which ones were the primary lipases which were problematic in mAb-based products? And using AlphaBase, we got this list of 8 different lipases, which we then could set up a MRM assay to detect the different lipases with. And this was something which was successful for this client, as we were able to detect that actually was LPL, which was causing this polysorbate degradation in their final DS batches. So another thing is, we were approached by a client which was having which was uncertain if we would be able to find a specific protein, and this specific protein was CCL2, which is a small cytokine which is known for recruiting immune cells to sites of inflammation. So it's immunogenic, and so you could say that this is quite a nasty host cell protein that you don't really want in your samples. And one of the reasons why they were concerned is because it's quite a small protein with a very high molecular weight. So they were afraid that mass spectrometry wouldn't be able to capture or identify this protein. However, because we have this database, instead of having to do some in silico analysis, which you also could do, you could just simply ask AlphaBase, have we seen this protein before? And here we give a clear answer very quickly and say, we have seen it in multiple samples ranging from harvest to DS. We've seen multiple different peptides across this protein. And then we could also check, how does this fit with what we would expect to see? Does it cover the areas we would expect, or does it cover things where we shouldn't be able to find this? So as you see here we have the protein sequence. This is the signal peptide. We don't expect to find that. All the white ones, or the white amino acids here, we would expect to be able to find, I guess. These ones, they're end-linked glycan sites. Those we would not expect to find. So, but all the green ones here, those are the ones we found. So that confirmed that for us, we were able to find this, and this is something that the client didn't have to worry about. So now I've shown you some of the things we can ask this database, just kind of like the tip of the iceberg. And with all of this data and the increasing amount of this data which will be generated, we need to ensure both the quality of the data, but also the quality of the analysis. So how can we do this with this database? So the way we approached this process was actually to create an automated quality control pipeline using machine learning. And this is kind of like the dream setup which we were hoping for, where we would have our client samples and some system suitability samples to measure the variability or problems in the instrument. And the client samples would have some internal protein standards as we're already using. Then we would generate the data, and then an analysis would be performed to check if the data is good or bad, and then it could be sent directly for troubleshooting if there was any problems. So we set out to do this, and for this, of course, we needed these system suitability samples and internal protein standards. We needed a standardized and automated high throughput setup, otherwise this just wouldn't make sense to do. But this is what AlphaLISE has, so this is something we could do. And then we need an AI-based QC system. So this is something we set out to develop. So to develop this, one of the key things is actually understanding the data and understanding what you actually want to get out of it. So you need to collect all of this data, and you need to annotate it based on what you actually want to get out of it. So you really need to think about what is good, what is bad, what do I want to for this machine learning model to actually classify based on. So we set up a lot of different parameters and really thought a lot about it. And then we started annotating all of these samples which we're running. So we actually spent a lot of resources on this. And one of the main important things about this is actually the consistency of these annotations and having some clear guidelines for how you're actually going to do this. And then safety in numbers, you really need to get a lot of data for this. This is one of the key things for training these models. The more data, the better. So how would you actually set up one of these machine learning pipelines, you could say? And this is just kind of like a general overview, just to give you an idea of how this is done. So we have a training phase and a prediction phase where you're actually using this model. And for the training phase, you start with your data and you want a lot of it. Then you need to do some annotation. And like I talked about before, this is actually one of the really important aspects of it and takes about 90% of the time. Then we have some feature extraction, getting out the important features and removing the ones which are irrelevant, just to save time. Then we train a machine learning model for the different things we want to annotate or to extract and classify based on. And then we have our trained models. In this case, I have 4. And there's a lot of validation and testing of these to make sure that they actually are performing as they should and that there aren't any overfitting or whatever happening in this case. So we ended up with an accuracy of between 92 and 97%, which actually is quite high. And this is kind of attributed to the high level of annotation, but also the high level of data we have been generating. And when you want to apply this in practice, then you essentially just provide new data to it, and you extract the important features, you introduce it to your trained models, and then you get your predictions. So it's actually, the latter part is simple, but everything else is quite complex. So how have we actually implemented this at AlphaLISE? So the first part is data generation, of course. So we have our client samples where we have our internal standards we're looking at, and then we have our system suitability samples where we're looking at BSA or a more complex sample like K562. We have our quick QC segment, which does a very fast evaluation where it converts the raw files and then searches them very fast and takes about 5 minutes. So you get a very, very quick answer if there's any problems on the instrument, so you can actually stop it from running any more customer samples. Then we have the thorough QC, which takes a little bit more time and goes through our standard analysis pipeline, and then does an evaluation on this. This is all collected in a report, and it's actually quite intuitive because you either have a red, orange, or a green light here. So as long as you're not colorblind, then you'll be okay. And it just indicates if the system or the samples are good or not. And then also, if you want, you can dive into the details and figure out what is the problem with this, if there is a problem. So it has a lot of different, figures and plots to demonstrate the different elements of this analysis. And it also has a overall score indicating the level of quality of that specific sample. So then you have very clear indication of how your sample has performed. So an example of one of these plots is this multidimensional scaling plot. Now, the nature of this plot is a little bit complicated, so I will spare you from the details. However, overall, this is a way of visualizing the differences between the individual samples. So this is kind of what the machine learning model actually sees, and it visualizes for the user even the smallest differences in these samples. So I'm sorry if you're colorblind, but in this area here, we have the green samples, which are classified as good. In the middle here, we have all of the potential not good, not bad samples, and on the left over here, we have all of the bad classified samples. So it's quite a clear separation of this, and it allows for some segmentation and classification of where the different problems are. So you can kind of get a visual analysis of this as well. And you can also see where your actual samples are placed based on this. These are green and obviously good because they're clustered with all of the good samples. And these are obviously bad because they're classified with the bad samples. But then you can always dive into further, why is this a problem? And this is something where we have readily implemented here at AlphaLyse. So why should you even have a QC tool like this? So firstly, a machine learning based quality control tool takes a lot of time, and you need to really think about what you actually want to get out of this. But the outcome of it is that it actually removes this subjective evaluation, which really can be a problem, because you can almost always find something wrong with a mass spectrometry or mass spectrometer if you're actually looking for it. But it might not mean that there's actually anything wrong. So it removes this subjective evaluation and is based on real data and what you should be finding, and not if you've not depending on different experts. So it also removes this need for having experts looking at the data constantly, because I don't know about you, but when I was at the university working with mass spectrometers, running my samples, I would not check all of the system suitability tests. I would check maybe 2 or 3 of them, but I wouldn't check all of them. And this system automatically checks all of them and gives you a notification or an email if anything goes wrong. So it's completely different level here. And it's easily interpretable and does not require any extensive training. It has this traffic light indication of the quality of the sample. So it gives a very clear indication of if anything is wrong or not. And if you really want to, you can always dive into all of the extra details if you want. So overall, this has so far reduced downtime for us, and thereby increasing throughput, because we do not hesitate based on this, because if anything is wrong in these samples, then we are able to quickly identify it and prevent this. And if nothing's wrong, then there's no reason to abort anything. So this overall enables a robust and reproducible LC-MS analysis. So what are some of the take-home messages from this? So overall, this new USP general chapter will generate a lot more data than on HCPs than ever before. So this means that we actually have to almost have an obligation to use this data and learn from it, because so many people will be generating a lot of data on this, and it would be a shame not to share and use this, and be able to improve biopharmaceuticals based on this. Another thing is individual HCP documentation enables data-driven decision making. So, for example, as we saw with AlphaBase before, we were able to take some decisions based on the data we have available. So we could make some clear answers on what actually is available and what we can do. Have we seen this before? Have we not seen this before? Is this a high level of host cell protein or not? And thereby compare between different projects and samples. It's important to have different system suitability and quality control steps in place to monitor your system and make sure that this analysis is performing correctly, because the quality of the data is very important if you want to use it in a database like this. And then something which is coming up, development of tools for improved identification and quantification of host cell proteins using machine learning. And this is something which I really think all of this data will enable. It will take a lot of time, but it's something which will vastly improve this analysis if used correctly. And I think that's it for me.
Mm-hmm. Thank you very much, Victor. So we can understand that this is a quite complex topic, and both difficult to understand, but also opens us a lot of perspective for how it can be used. So I'll just let you catch your breath while let me just see if I can go back to our live broadcast here and stop presenting. Okay, it's turned off. So yeah, if you have questions for Victor, then please type it in the chat, and we will go through your questions. So first, Victor, I will start off while people are thinking about questions. So I know you have been trained in mass spectrometry from our local university, a proteomics group, which is quite famous for their proteomics research, and they have a lot of mass spectrometers and a lot of experts in running very complex samples. So what are the problems when you work in a mass spec lab and you try to collect good data on your samples? What are the challenges as a mass spectrometrist?
Well, there are quite a lot of different problems, which overall you just usually consider getting the best results for that one analysis when you're at the university setting. You don't necessarily always consider the being able to reproduce this again. So you'll do your best for optimizing it for this one run, and you'll make sure that the instrument is running at optimal performance before you actually put your samples on it. But so it's generally quite difficult actually to reproduce these results again. At least that's what I've experienced in my own personal time there, and also making sure that the instrument is running properly, and also then identifying what was the problem. So that was one of the things I spent a lot of time on. Also looking at these individual system suitability tests, or for example the BSAs or a cell lysate you're running in between your samples, that would take a lot of time if you were going to check all of them. And often, personally, I wouldn't check all of them. I would check maybe the first few ones before to make sure that the system was running as it should to my specifications, and then I would maybe check some of the last ones, but rarely any of the ones in between, unless something had gone wrong, which I could see in the data. So you wouldn't necessarily identify if anything was off during this analysis.
So basically, when you acquired the data, you wouldn't know if the data were good or bad. It was not until you did a deep dive into the data analysis, maybe you would find out if you ran duplicates or triplicates that there was something wrong in one of them. So whereas the tool you demonstrate is that you get actually the answer on the fly. Is this analysis red or green?
Yeah, so it will let you know if anything's gone wrong before, not before it happens, but kind of in a preventative measure to ensure that you don't run any additional samples. Because if you only identify that there's a problem once you start analyzing the data, maybe a week or two after when you have time, then a lot of time will be wasted. And if you, at least back when I was at the university and running my samples, then if I had looked at my samples right away, then I might have had time to put an extra sample on to rerun this to make sure that I would have my data in time.
Yeah, so different type of question here is on the quantitative analysis on the ppm calculations. So do you use all the 7 standards to normalize, and maybe you can give some details how you use the 7 standards and how do you make the ppm calculations?
Yeah, so overall we actually do use all 7 standards as it is right now. We use them and we calculate a median response factor based on these 7 protein standards. And this means that it actually, if anything goes wrong with one of the protein standards, then it will be accounted for by the other ones. So this creates a very, very robust quantitative, quantitative approach. And then you use this median response factor compared to the measured intensities of the target host cell proteins, and then you get the quantified ppm from this. I don't know if I should go into more details or if that's sufficient.
That's completely fine, Victor. So I have another question in the same category. So these 7 proteins you use as standards. So why did you select these? Why did you select 7?
I think that might be more of a question for you, Ivan.
Yeah. So maybe all these HCPs, they behave in a different way. So it's to present this variability of individual proteins that we selected a number of them. And when we have 7, it enables us to select the median one, the one in the middle. So actually we're looking at the spread of how these proteins they perform. So they've been selected for different criteria. First of all, we could get them in pure form. We could, they are not recombinant proteins, so we don't have expression system proteins from CHO and E. coli, for example. So they're pure and well characterized, and we have the known amounts. So I know that the US Pharmacopoeia, as part of this chapter, are also working on standards that you can use for the different approaches. So they are giving presentations that will share more about this in the coming months. So you can actually buy these standards in the future from the US Pharmacopoeia. So let me just read some more questions. I think there's a bioinformatics question here. Hi, it sounds amazing with the database system. In your LinkedIn post, you mentioned Shiny, which was used to build a user interface. Is this user interface for extracting, looking up data also plotting? And how does the database metadata allow you to reproduce the experiment?
So we have quite a few different Shiny apps, and we haven't created a Shiny app for the database curation because that's something. The Shiny apps are generally for the non-bioinformaticians, as this creates a user-friendly way of using these processes or analysis. And at the bioinformatics team, we generally just use different, not scripts, if you can call it that, and for curating all of this data in an automated fashion, and then it automatically updates this database following some annotation of metadata. And all of this metadata is something which is collected from all of the different analysis, and it's kind of based on different types of samples and the buffer components, expected host cell protein levels, and all these kind of things. So we get a lot of different insights into what actually is these samples consist of, and so it's not so much for reproducing the what was the question, last part? I can't see it anymore. Yeah, but overall, it's the metadata isn't necessarily used for recreating these analyses, as we already have the ability to recreate this, because the method itself is a general and robust method, so we don't really need to change anything for the specific sample. So it's overall the metadata is mainly for asking questions to the database and for all filtering and things like this.
So Victor, we're running a little bit over time here, so not to take up too much time of our audience. So I think I have one last question looking into the future. If we look into the crystal ball here for HCP analysis, so now we have the new USP chapter 1132.1 describing 3 different mass spec methods to identify and quantify HCPs. You have demonstrated the analytical methods to acquire these data very reproducibly, and now we have set up a database to collect all this information and query it in different ways. If we look into the future for biologics development, what will these new technologies and these new tools, what will it do for drug development? What will it do for the CMC responsible for developing a new biopharmaceutical? How can this information and new technology, how can be used to improve safety and efficacy of drugs?
Yeah.
Could you give us some thoughts on that?
Yeah. Overall, I would say I think it's kind of using all of this data is going to be helping towards creating a kind of a benchmark in how we look at host cell proteins. So with also the new general chapter from USP coming out on how you actually quantify these different host cell proteins with some different methods, this will create a standard of how you're actually supposed to do this. And this will enable comparison between, I hope, different labs. So we actually can also, I hope, share information and get a compare different levels of these host cell proteins, also based on what is actually on the market currently, because then we can link that to different clinical trials and see what different effects these different patients have had based on the different host cell protein levels. So we'll be able to get a lot more information on why actually which host cell proteins actually are a problem, and at what relative level to these, for example, protein standards, are they actually a problem? Because if we all used the same intact protein standard for this quantification and used the same method, then based on the physiochemical properties of these proteins, then it would be comparable. So then we'd be able to compare across all these different, let's say, laboratories and see what level are these host cell proteins actually a problem. So we could get so much information out of this, and it would greatly improve the risk assessment and the assessment of these host cell proteins for the future of biopharmaceuticals.
Mm-hmm. Thank you very much, Victor. So I'm afraid we don't have time to answer all of the questions. So after the webinar here today, so we will follow up in writing and contact you for those questions listed here. And also, if you have questions later on, please don't hesitate to contact us. Then Victor and I will be happy to answer you by email. So with that, I would like to thank you for your attention at this webinar and for your good questions, and especially Victor for giving this nice presentation on this very complex topic. So thank you very much all. Bye-bye.
Bye.